Good evening, everybody. Just join fast. I need all of you. Join fast. So let's start our lesson. Today, biological classification. Another very important chapter. Let's crack it. So the biological classification, starting with Aristotle. Aristotle's classification. Aristotle was the earliest to attempt a more scientific basis for classification of organism. He classified plants into trees, shrubs, and herbs, and animals are classified into two groups, that is, I think you all know this one. So those with red blood, enema, and without red blood, that is anima. Enema and anima. So Aristotle, the earliest to attempt the more scientific basis of classification, he used the simple morphological character. In case of plants, he treated only the morphological characters and classified the plants into trees, shrubs, and herbs. And animals are divided, that with red blood, anima, that is enema and anima. Enema with red blood, anima are without red blood. Now, the earlier classification system It include the bacteria, the blue-green algae, fungi, mosses, ferns, gymnosperm, and angiosperms. So they are all under the plants group. Because all these components, they have cell wall. So it brought together the prokaryotic bacteria, and the blue-green algae, that is, we can say cyanobacteria, with other groups which were eukaryotic. It also grouped under, that is, Chlamydomonas and Spirogyra are placed together under algae. So Chlamydomonas and Spirogyra so they are under unicellular and multicellular group. Hello Devjit, good to hear. So now the classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants. So now after two kingdom classification, the three kingdom classification, then four kingdom classification, and lastly the most accepted one, that is five kingdom classification. So you can remember who proposed this. So can you tell me who proposed the five kingdom classification? Yes, Robert H. Whittaker. So before moving to next topic, just we have to recapitulate the different classifications. That is two kingdom. Two kingdom classification. It is given by Carolus Linnaeus, that is in 1758. The famous Swedish naturalist. The two group, that is Kingdom Plantae and Kingdom Animalia. But the two kingdom classification shows lot of demerits. As the euglena, some of the other organisms exhibit characters of both plants and animals. Bacteria neither match up the plants for their animals in cellular structure. Fungi is being non-photosynthetic is grouped under plants. The sponges are sessile in nature but placed in Animalia. So these are the different faults and demerits of two kingdom classification, and that bring the three kingdom classification. And the three kingdom classification was proposed by, tell me, everybody participate. Give your answer in chatting box. Ernest Haeckel. Yes, you are right. That is 1866. So the third kingdom that is proposed by Ernest Haeckel, that is Protista. So that including, very important, unicellular eukaryotes. Like the protozoan and algae. But the three kingdom classification also faces the problem is heterotrophic bacteria and fungi are placed along with autotrophic algae. So the fourth kingdom classification came. And the fourth kingdom classification is proposed by, yes, very good, Ashish, the Copeland, that is Herbert Copeland. And that is during 1956. In this classification system, bacteria and microbes are included under Monera. So Monera is introduced. Now the four kingdoms are Monera, Protista, Plantae, and Animalia. So the Monera include the prokaryotics organisms. Protista include unicellular eukaryotes. Plantae, the photosynthetic group, and Animalia, the non-photosynthetic. So these are the four kingdom in Harvard Copeland during 1956. But there are also demerits that fungi. See, the fungi is a non-photosynthetic but grouped under Plantae. So it's a fault. That's the emergence of the five kingdom classification by Robert H. Whittaker, and that is 1969 five kingdom classification. So he introduced fungi into separate group. So the classification was followed in almost the textbooks, widely in different textbooks. So the five kingdom classification by Whittaker, so it has some merits, lot of merits actually. It places prokaryotes, the prokaryotes under the Monera. It is one of the merit. It placed the unicellular eukaryotes into the kingdom Protista. That is another merit. It elevated fungi to the level of a kingdom. It separated fungi from the plant. So it is based on the characters which evolved from every organic world. So cellular organization and the modes of nutrition. So now we are moving in detailed structure of five kingdom classification. Look at here. This is the prime thing we have to understood in very clear concept regarding this. Look at here. So, first thing, what are the criteria? So, this is the first criteria, that is complexity of cell. The complexity of the cell, that means they are either prokaryotes or eukaryotes. They under prokaryotes, the kingdom Monera, and their chemosynthesis is there. So now the second category, that is complexity of organism. So they are either simple or complex, simple or unicellular, and complex or multicellular. Here the kingdom Protista is there. So Protista is under simple unicellular eukaryotes. Then they are bifurcated into Plantae, Fungi, Animalia. Now the third category of division, that is model of nutrition. See, the model or the mode of nutrition, They are autotroph because they are capable of photosynthesis. They are heterotroph, the fungi, so their mode of nutrition is absorption by their mycelium and hyphae. And the animals, they are heterotroph and their mode of nutrition is ingestion. The fourth category of classification, that is ecological role. So the plants are producers, the fungi are decomposers, and animalia, they are consumers. The fifth category, that is the fifth category of classification, that is direction of evolution. That's the phylogenetic relationship, starting from the prokaryote, then eukaryotes, then unicellular eukaryote, then multicellular, then plantae, then fungi, then complicated animalia. So that's the phylogeny, the fifth category of classification. So the complexity, number one. Number two, complexity of organism. Number three, model of nutrition. Number four, major ecological role. And number five, the direction of evolution, that is phylogenetic relationship. That's very important. What are the criteria of five kingdom classification? So take a screenshot. It's a very important thing to remember, and all the questions that you face from this one, the Whittaker's classification, so that provide the answer key of all questions of five kingdom classification. So are you taking the screenshot? That's very important to remember. Now, so complexity of the cell, they are prokaryotes and eukaryotes. Complexity of the organism, they are simple or complex. Mode of nutrition, autotrophic, heterotrophic, and major ecological role, producer, decomposer, and consumers. So keep it into mind. Monera, Protista, Plantae, Fungi, Animalia. So now, moving to the next part of our lesson. So five kingdom classification, though there are a lot of merits, but there are some demerits also. Some demerits are, number one, the kingdom Monera and Protista are still heterogeneous group. That means both photosynthetic and non-photosynthetic walled and wall-less organisms are there. So we can say they are heterogeneous group because photosynthetic autotrophics are also there. Non-photosynthetic groups are also there. Walled components and non-walled, that means wall-less organisms are there. Another demerit, we can say, the phylogenetic relationship particularly of lower organism, are not fully reflected here. For example, we can say the certain green algae and some photosynthetic bacteria get hydrogen from sources other than water, but these are assigned to the different kingdoms. A distinction between unicellular, in case of unicellular and multicellular organism is not possible in case of algae. So due to this unicellular green algae, that means unicellular green algae, what's the example? The Chlamydomonas, the Volvox. These unicellular organisms have not been included in the kingdom Protista. They are not included in Protista. Viruses, another demerit we can say, that viruses are not included in this system of classification. And archaebacteria differ from bacteria in structure, composition and physiology also. So they also not separated in this kingdom of is 5 kingdom classification. So these are the problem related with 5 kingdom classification. So to overcome this problem, again the another introduction of classification is there, that is the 6 kingdom classification or 3 domain system. The 6 kingdom classification, and this system is proposed by, who proposed this? That is Carl Woese et al. And his associates, 1977. So in this system, kingdom Monera is divided into, so Monera is bifurcated into archaebacteria and bacteria. So now they are called archaea and bacteria on the basis of microbial cytogenesis. So the kingdom are, so what are the 6 kingdom? That is archaebacteria, eubacteria, Protista, fungi, plantae and animalia. See the 6th kingdom, 1, 2, 3, 4, 5, 6. So here the separation from the previous one, that is Monera is bifurcated. Monera is bifurcated into archaebacteria and eubacteria. Now the archaebacteria is called archaea, and eubacteria is called bacteria. So these are the modifications. So that's the Carl Lewis proposed three-domain system in biological classification. The domains are archaea, bacteria, and eukarya. These are the first domain, second domain, third domain. Three-domain system of classification. So next, in modern days, so the science is not an idle subject. So it always moves further and the development of research is going on. So the seventh kingdom is also arrived. So seventh kingdom, the system of classification is proposed by Ross in very recent 2 and 5. So he recognized Archaebacteria and Eubacteria as separate kingdom, named them Protomonera and Monera, respectively, again under the separate super kingdom Archaebacteriaceae and Eubacteria, and added the seventh kingdom Myxomycophyta of slime molds under super kingdom Eukaryotes. So that is the seventh kingdom, that is Protomonera, Monera, Protista, Myxomycophyta, the fourth one, then the fungi, plantae, and animalia. One, two, 3, 4, 5, 6, and the seventh kingdom by Ross in 2002 and 2005. Now, we are moving to the different groups and their important characteristics. Starting with kingdom Monera. So everybody like, share and subscribe to get the latest information. Stay with us. Number 1, the kingdom Monera consists of prokaryotic organism. Number 2, Monera are unicellular. And cosmopolitan. Many of them are decomposer. The nuclear material remain freely suspended in the cellular cytosol, and this nucleus is called nucleoid, or this is also called incipient nucleus. Very important. What do you mean by incipient nucleus? Mark it. So next, they lack membrane bound cell organelles like mitochondria, nucleus, chloroplast. They are lacking here. The plasma membrane invaginates to form the respiratory structure that is called mesosome. Very important. What is mesosome? Remember this one. And they have, besides mesosome, the another important, the photosynthetic structure, that is chlorosome. Mesosome, respiratory structure, and chlorosome, that is photosynthetic structure. They contain respiratory pigments. So these are consist of the respiratory pigments. And chlorosome, they contain photosynthetic pigments. Both are associated with separate pigment. One is respiratory, another is photosynthetic. Chlorosome are present in some autotrophic bacteria. So the chlorosome is present in the green sulfur bacteria. Very, very important. Green sulfur bacteria. Keep it into mind. Very important concept. Mesosome, respiratory pigment, chlorosome, photosynthetic pigments, and chlorosome are present in the green sulfur bacteria. So everybody, is it clear? So give your response. Is it clear to you all? Mesosome and chlorosome. Next, the cell wall are principally made of peptidoglycan. So what is peptidoglycan? So peptidoglycan, a heteromolecular fibrous macromolecule which is a combination of amino acid and sugar that surrounds the cell like a network. So the cell wall is composed of peptidoglycan and teichoic acid. Teichoic acid and a lipoprotein. What's the name? That is Burns lipoprotein. Burns lipoprotein. Very important, the cell wall composition. Next one, the moneron. So moneron cells are 70th type of ribosome. Their ribosomes are 70th type. Next, one, the monerons multiply to reproduce asexual. They lack true sexual reproduction. They are incapable of gametogenesis, but genetic combination may occur. That's very important. Again, I am repeating, they are incapable of gametogenesis, but genetic recombination may occur, and that is due to conjugation, transformation, and transduction. All are there. Conjugation, transformation, and transduction. Now, the cellular division involves what type of cellular division is there? Cellular division is amitosis. Yes, Ankita, you are right. Amitosis. They lack true sexual reproduction. So, which amitosis does not involve the mitotic machinery or spindle apparatus. So, in amitosis, you know this one, that is the direct cell division, so no mitotic apparatus are formed here. The principal hereditary matter we already described, that is the circular DNA, that is called the nucleoid. So there may be other accessory circular DNA molecules present in the monomeral cell. So other circular DNA other than nucleoid, that is called plasmid, and episome. Yes, Ankita, you are right. Episomes. Plasmids and episomes. The major monera, that is Archaebacteria and Eubacteria. And Eubacteria is further divided into bacteria and cyanobacteria. In Archaebacteria, the major types are, three major types are methanogens, halophiles, and thermoacidophils. So, do you know the methanogens? They are obligate anaerobes. In the biogas fermenter, the bacteria produce methane. That is methanobacterium, methanococcus. Give example. Methanobacterium. Yes, Supriyo, very good. Gut of ruminant stomach. The ruminant stomach of the herbivores are associated with methanobacteria, methanococcus. The halophils. The halophils, they are called salt-loving bacteria. Salt-loving bacteria, as they are found in the environment with a high salt concentration. And the thermoacidophils, live in extremely acidic environment. That means where the pH is less than 2, pH less than 2, that have extremely high temperature. The thermoacidophils, that means up to 110 degree Celsius, and they found in hot sulfur spring also. So high range of, so they are cosmopolitan origin. So these are the prime characteristics of Monera. So everybody keep into mind the important characteristics of Monera. The prokaryotic organism, unicellular and cosmopolitan, they are decomposer, nucleate and incipient nucleus, that is called lac membrane of cell organelles, mesosome and chlorosome, peptidoglycan, teichoic acid and bond lipoprotein, these are present here. 70S type of ribosome are there. Cell division is mitosis. Circular DNA is nucleoid. Other than circular DNA, they have plasmids and episomes. They are divided into archaebacteria into methanogens, halophils and thermoacidophils. So everybody, is it clear to you all regarding Monera? So everybody, yes, thank you. So the next one, that is Protista. The next one, that is Protista. We are moving to the next page for kingdom Protista. Kingdom Protista includes, there are all unicellular, eukaryotic organisms are there. So phylogenetically, kingdom Protista act as a connective link between prokaryotic kingdom Monera and the complex multicellular organism. That means Protista is a connective link between the unicellular and multicellular organism. Very important thing to remember. These are microscopic unicellular eukaryotic organisms, cosmopolitan habitat. Many of them are formed planktonic biomass. Planktonic biomass. So, cell with one or more cell developed nucleus, presence of well-developed membrane-bound organelle, their mode of nutrition, they are parasitic, ingestive, photosynthetic, saprophytic and mixophytic. All types of mode of nutrition are there. This group includes the photosynthetic phytoplankton, non-photosynthetic zooplankton. So the planktonic biomass, that is photosynthetic phytoplankton and non-photosynthetic zooplankton, both are there. Some of the individuals have cell wall like peripheral covering. They have peripheral covering that is called exoskeleton. Very important. Note down this one. Sometimes covered by the soft pellicle. But most of them are with plasma lemma only. They have pseudopodia. They have flagella, that is 9+2 arrangement, microtubular arrangement. That is the flagella. And some of them having cilia. As their locomotory organs. They have contractile vacuole. What is the function? What is the function of the contractile vacuole? Any one of you? So contractile vacuole that is present in the cytosol that helps in osmoregulation. Osmoregulation. Reproduction occurs generally in asexual method. Mostly these are common to us, the binary fission, multiple fission. So binary fission and multiple fission. So fission are always under favorable condition. These are under favorable condition. But the budding, encystment, sporulation, these are the budding, encystment, sporulation, these are always in unfavorable condition. Keep it into mind. Sexual reproduction executed through conjugation. So conjugation is there, or by the fusion of nuclei. Another mode, that is syngamy. Another important fact of Protista, very important, two types of life cycle. The life cycle of Protista are mainly of two types. One is zygotic meiosis and another is gametic meiosis. Zygotic meiosis in dinoflagellates and in cellular slime molds. Note down this one. The zygotic meiosis is present in dinoflagellates and cellular slime molds, and gametic are present in majority of the protozoan, the protists, the diatoms, the acellular slime molds. So very important in case of slime mold. So cellular slime molds show zygotic and acellular slime molds show gametic meiosis. So the next part, that is fungi. So everybody stay live. The lesson is not completed, so where are you going away? Take tea or coffee and stay alive. Kingdom fungi. So these are the unique kingdom in the heterotrophic organism. They are cosmopolitan. They grow in warm and humid places. They found in the mold or bread or rotten fruits and mushroom, the toadstools. Some fungi are source of antibiotic also, the penicillium. Some unicellular fungi are used to make bread and beer. Other fungi, they cause diseases of plants and animals. That is the Puccinia graminis triticii. Their bodies have thread-like structure. These are called the hyphae. And the network of hyphae is called mycelium. The fungal hyphae are mainly of two types. That is cenocytic and the septate. Cenocytic are aseptate. Most of the fungi are saprophytes. Some are symbionts. That is, in case of lichens, that is the association of fungi and algae. They are symbiont. And there is another example, that is mycorrhiza. Here the mycorrhiza is the fungi is associated with roots of higher plants. Yes, Pooja. So reproduction, these are vegetative propagation by fragmentation, fission, budding. They show the conidia as asexual reproduction, zoospores, sporangiospore. They have all oospores, ascospore, basidiospores. So these are the distinct structure of their fruiting bodies. In fungi, we found that there are 3 sexual cycles. The 3 sexual cycles are there. That is plasmogamy, karyogamy, and meiosis. So 3 sexual cycles in there. In plasmogamy, the fusion of protoplasm between the 2 motile or non-motile gametes. In karyogamy, the fusion of 2 nuclei, and meiosis in zygote to divide the haploid spores. The fungi on the morphology of mycelium and mode of spore formation, they are classified into 4 categories. So can you give the example? What are the fungi classification? That is phycomycetes, ascomycetes, basidiomycetes, and deuteriomycetes. Yes. So phycomycete is the lower fungi. The mucor, the rhizopus, that is called bread mold. Albigo, that is parasitic fungi on mustard. Ascomycetes called sac fungi. So they are called sac fungi. Aspergillus, claviceps, neurospora. Under Ascomycetes, the neurospora is used in the all biochemical and genetic work. Basidiomycetes, these are mushrooms. They are called puff balls. Some plant body, they are living as parasite, the rust disease and smut disease. Their fruiting body is Basidiomycetes, the basidiocarp. So basidia are arranged in fruiting bodies, that is basidiocarp. The agaricus, Eustylego, Puccinia, these are under Basidiomycetes. The Deuteriomycetes frequently asked question, they are also called imperfect fungi. Because only their asexual and vegetative phase is known. Their sexual phase is yet to known. They reproduce through conidia, the alternaria, coleotrichum, trichoderma. These are the examples. So another part, that is plantae. We know this one regarding plantae. Plants are eukaryotic, chlorophyll containing, algae, fungi, bryophyte and pteridophyte, gymnosperm, these are there. We discussed this part in plant kingdom. And the animal kingdom, we divide this group and separately we discuss in the animal kingdom part. The another important thing we have to discuss in this part, that is the viroids. So, who give the name viroid? Can anybody tell me? That is T.O. Denier, 1971. Yes. So, it is a new infectious agent, was smaller than virus and caused potato spindle tuber disease. It is found to be a free RNA. It is lack of protein coat and found that present in virus, hence their name is viroid. No protein coat is there. The RNA in the viroid, so viroid's RNA are low molecular weight. Low molecular weight. After viroid, the prion. So an abnormally folded protein which transmits certain infectious disease. Remember this one. Prions are almost same size to virus. So the important disease caused by prions, That is BSE. Can you tell me the full form? That is bovine spongiform encephalopathy. Commonly called, this is called mad cow disease. And the another disease, CZD. That is crack-Jacob disease in humans. So that's the human disease. So we are almost completed this part in very brief way. So next day we completed the another one. So next day we start the another important chapter, that is plant kingdom. Then we describe the part which we are not discussed today, that is plant kingdom. And the fourth day we discuss the animal kingdom. That will complete the whole part of the first unit. And we also practice lot of question on the fourth day in MCQ. So today we conclude our lesson. So bye everybody. Keep sharing and subscribe the channel and share to your friends. Those are biology lovers. Till then, bye everybody. Good night.